a fantastic day we've been having. We're into session 4, our last session for the day, and continuing on the great conversations of all of our speakers. So without further delay, Dr. Eric Smith joins us. Dr. Smith is the first of our speakers in session 4 concerning COVID-19 and the brain. Dr. Smith investigates vascular contributions to age-related cognitive decline and dementia, including stroke and stroke outcomes. He studies people from the general population, as well as patients with clinical cognitive disorders and cerebral amyloid angiopathy, a vascular disease caused by beta amyloid. Dr. Smith's research is funded by the Canadian Institutes for Health Research and Brain Canada, as well as other sources, in collaboration with a team of vascular specialists in the Canadian Consortium on Neurodegeneration in Aging, including both basic scientists and clinical researchers. He is identifying new ways of treating people with cerebral small vessel diseases and testing them in clinical trials. Dr. Smith, thank you for joining us.
Thanks so much, Jordan. And my pleasure to speak to the group here. I'm going to talk about, my main objective is to present a project that is an ongoing project, and I'll explain the rationale and study design. It's a study looking at the effects of exposure to COVID-19 on brain structure and cerebrovascular lesions, embedded within the Canadian Longitudinal Study on Aging, which is an open science data platform for Canadian and international researchers that everyone in Alberta can access. So without further ado, why don't we get going? We can go to the next slide, please. And these are my disclosures. So I've done consulting for these companies on trials related to vascular cognitive impairment. I receive royalties for writing a chapter on diagnosis of vascular dementia for Up to Date, which is an online medical textbook. I'm not an infectious disease expert, I should disclose. And I'm a co-PI of this study that I mentioned to look at the effects of COVID-19 on the brain, funded by CHR and the Western Brain Institute. I'd like to thank my colleague Landon Perlett, who's a neurologist and clinical fellow in Calgary, who drafted the first version of some of these slides. Thanks. Next one, please. So I'll give a brief overview of COVID-19, talk about some of the brain symptoms that have been described, what's known about the neuroimaging of COVID-19, and the rationale, objectives, and design of this research project. Next slide. So COVID-19 is coronavirus disease 2019. It's a new human disease caused by severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2, which is a single-strand RNA virus. It includes 27 proteins. One of the key proteins is the spike protein, shown in this kind of schematic on the left. The spike protein is on the outside of the virus particle, and it engages ACE2 receptors, and it's the key protein that engages with the cell that allows infection of the cell. ACE2 receptors are expressed widely on respiratory epithelium, but they're also found in fairly high concentration on human endothelium. So infection of the vessels is one thing that you see in COVID-19, and may be one explanation for some of the brain symptoms that we see through infection of the cerebral vasculature. And that's one of the things we're investigating in this project. Next slide, please. So the symptoms of COVID-19 are primarily respiratory and also general viral symptoms. So fever, chills, and cough, shortness of breath, fatigue, and aches are the most common symptoms. Headache is pretty common too. Anosmia or agusmia, loss of sense of smell and taste are quite common. And also symptoms like sore throat and runny nose, nausea, vomiting, and diarrhea. Next slide. Most cases are mild, but up to 20% of cases can lead to emergency room visit or hospitalization. About 5% of cases, the patient is critically ill. These are numbers from the early days of the pandemic, before there was any immunity in the population. So this may have shifted a little bit toward the milder cases. And of course, in the very mild cases, the symptoms can be so mild as to be nearly unnoticeable or even asymptomatic. Most of our understanding of the neurological effects of COVID, though, have been limited to the patients that have been hospitalized with a number of tests. So one area for further understanding is increasing our understanding of the effects of symptomatic COVID on an outpatient basis in the community. Next slide. So I'll talk about some of the effects of COVID-19 on the brain and on neurological symptoms. Next slide. So this is from one of the early papers cataloging symptoms in hospitalized patients. So they found dizziness and headache quite common in about 1 in 6 and 1 in 7. Some patients had elevation of creatine kinase, which is a symptom of muscle breakdown. To a neurologist, the muscular system is part of the neurological system. 8% of patients had impaired consciousness or delirium. Anosmia seen in 5%. Stroke seen in 3% of hospitalizations. And neuropathy, ataxia, seizures. Next slide. And in patients that are hospitalized with COVID or critically ill with COVID-19, there are a number of different potential pathways by which the infection and the syndrome could affect brain function. So one intriguing possibility is a direct invasion of the virus of neurons. And I'm hoping maybe some of the other speakers will educate me more about this, but this is quite intriguing and suggested in some basic science work. There are also effects on the body that may have secondary effects on the brain. For example, there's very robust inflammation. There's peripheral organ dysfunction, including failure of the lungs with hypoxia, myocarditis with suppressed cardiac ejection fraction leading to hyperperfusion or formation of thrombus in the left ventricle, and embolism to the brain is another mechanism. There's often acute renal failure in the patients that are critically ill. There are cerebrovascular lesions that have been seen, potentially arising from direct infection of cerebrovascular endothelium. There are potential bystander effects of autoimmune attack on the nervous system driven by the robust activation of the immune system by the virus. And there's the effect of hypoxia, and then also the potential effect of treatments, including medications. Next slide. So for example, in hospitalized patients, it's not uncommon to see small brain infarcts, as seen in the MRI scans and panels labeled A to D. Panel D is a high-resolution flare, showing a tiny hyperintensity in this cortex representing a small infarct. And then the two panels at the bottom, E and F, show axial slices through the cerebellum, where there's a bright signal in the bottom left panel, indicating an area of DWI positivity, and the ADC map on the bottom right panel shows that it is dark, indicating restricted diffusion, so that's a tiny acute infarct in the right cerebellum. Next slide. Perfusion scanning in patients with severe COVID has shown that there is hypoperfusion in the brain, and this studies using fluorodeoxyglucose positron emission tomography, FDG PET, have shown also that there's hypometabolism in regions of the frontal and parietal lobes and cerebellum in patients with acute severe infection. Next slide. And a rapid review published in 2020, so now getting a little bit old in the COVID world, looked at 26 studies. Now, most studies, even till now, most studies of neuroimaging findings in COVID have been limited to case series from hospitalized patients, sometimes even case reports. At that time, they aggregated information from 361 patients, kind of clinically relevant, radiologically visible to a radiologist abnormalities were seen in 34%. Most commonly, there was evidence of white matter, hyperintensity, indicating edema on MRI, but other findings that were common included microbleeds, signs of small leaks of blood from the cerebrovasculature, and also infarcts and hemorrhages. Next slide. The neuropathology studies, and there are increasing numbers of studies. This was one of the early ones that included 18 patients autopsied at an American teaching hospital of patients, all of whom had delirium or confusion. They found hypoxic changes in the brain, and they detected by PCR low levels of viral RNA in most cases of patients that died acutely of COVID-19. Now, the levels of the transcripts, though, were about 1 million-fold lower than in the lung. So there was viral RNA present, but at much lower levels. It's not clear whether that viral RNA was in neurons or glial cells, or whether it was in the endothelium. They did not find evidence of viral particle staining on immunohistochemistry, so just either low level of viral infection or possibly contamination from blood viremia even. Next slide. And then this very recent publication in the journal Nature is the largest study looking at quantitative brain changes in outpatients with COVID-19. And one of the advantages of this study is that it was embedded within the UK Biobank study. The UK Biobank is a large science platform in the United Kingdom where over 100,000 people have had brain MRI scans. And they had a wave of brain MRI scans before COVID hit. And then they were able to select 401 cases who had caused in the panel on the left, the areas are indicate regions where there was more of this group. Oh, sorry, everybody. I think I got disconnected for a second. Am I?
We hear you now. You're back now, Eric.
Okay. All right. Good. Sorry about that. I'm on my university internet, so I guess University of Calgary. Sorry about that.
It's been worse in the last couple of weeks as students have returned.
I guess so. Yeah.
Yeah.
So I was just explaining what these red-orange blobs are and what they mean, right? So they're areas where the cortex has thinned more in patients exposed to COVID-19 compared to controls. And they found thinning in areas like the orbital frontal cortex and the parahippocampal cortex. These are regions that are connected to the olfactory system, and they suggest that it may reflect degeneration of gray matter from infection in the olfactory bulb, because there's a high concentration of virus in the nose that might infect olfactory nerves, and then that might affect olfactory cortex and connected regions. They also found areas of thinning in the anterior cingulate cortex. We'll go to the next slide, please. And then, of course, there's this long COVID problem where patients, even after recovering from acute infection, can have ongoing respiratory, neurological, or other symptoms, lasting for months, or we don't know how long, really, right? It's a very new phenomenon. And one of the largest studies here used medical record data and billing data from electronic health records in the UK to look among 236,000 patients with medical records. They found that incidence of neurological or psychiatric problems at 6 months after COVID was 33%, so basically 1 in 3 patients. And they compared these to controls, including patients who had other types of respiratory viruses like influenza. They broke the COVID-19 core down by severity into those who were hospitalized or not, and those who were in ICU or not. And about 3% of those were older than 65, and of 5% of those who had a delirium related to COVID went on to have a diagnosis of dementia within 6 months. Next slide. And next slide. Okay. And actually, previous slide, sorry. Thank you, yeah. And so these are Kaplan-Meier curves showing that the incidence of dementia and the incidence of mood or anxiety disorders seem to be higher in patients with a history of COVID-19 than patients that had other respiratory tract infections or influenza, suggesting there may be something pretty special about COVID-19 in the brain that's different than other respiratory infections and viruses. Thank you. Next slide. Okay, in the next few minutes, I'm going to present the design of this Canadian longitudinal study on aging study. And what we're focusing on are some of the effects that may be seen in non-hospitalized patients. Other questions, though, include like what's the source of this viral RNA in the brain? Is the virus replicating in brain endothelium? Is there a stroke risk after COVID-19 infection? What happens long-term to these patients that have neurological symptoms? And how does COVID-19 interact with pre-existing conditions and comorbidities to influence outcomes? Next slide. So the Canadian longitudinal on aging, next slide, please. It's a network of centers contributing information to Canada's flagship study on aging. These are the different sites where data is collected. One of them includes the University of Calgary. So it's a prospective cohort study of human subjects age 45 to 85 at enrollment. Next slide. The three leaders are Parminder Raina from McMaster University, Christina Wolfson from McGill, and Susan Kirkland from Dalhousie University. Next slide. And it's a research platform, so it's designed to collect data for other research to access and analyze. And access to the data is open to all qualified Canadian researchers, research institution, and also researchers worldwide. Next slide. It includes 50,000 people. They were aged 45 to 85 at baseline. 30,000 have inpatient visits that are called the comprehensive cohorts. 20,000 are followed by phone. And then the study began in 2015, and every 3 years, participants are reassessed. Next slide. There's a lot of data collected as part of this study, including from a brain standpoint, history of stroke and Parkinson's disease, as well as cognitive assessments with a 30-minute neuropsychological test battery. Next slide. Biospecimens are collected. There's also information on the environment and linkages to provincial health records. Next slide. So now I'll explain how we're using this study as a platform for study of COVID-19. Next slide. And our goals here are to understand better the epidemiology of COVID-19 among older Canadians. So blood samples were done to look for serologies for COVID-19 in 2020, kind of during the first wave of the pandemic. And this information, we also collected information by phone about history of COVID exposure in 28,000 CLSA participants. Next slide. So that's the questionnaire study. The seroprevalence study is the one where we've collected blood, and that's in more than 19,000 CLSA participants. And that began at the end of 2020. So the blood was collected in 2021, kind of during the second phase. Next slide. And then the brain study is where we're looking more intensively at, in a nested case control design in patients with and without history of COVID-19, somewhat analogous to the UK Biobank study, where patients are going to have brain scans, and they will look for structural changes and to see whether the prevalence of cerebrovascular lesions is higher in the cases compared to the controls. And so that launched in May of last year. It's funded by CHR and the Western Brain Institute. And we're looking, actually, the sample size is clarified now. We have 656, of which half will be COVID-19 cases and half will be controls. Mostly they are patients who have outpatient community-acquired COVID-19. And the PI is Teresa Leo Ambrose from UBC, and I'm the co-PI, and I manage the Brain Core Lab. Next slide. So we know that coagulopathy and vascular endothelial dysfunction occur in COVID-19 infection, at least the severe, more hospitalized cases. And this could lead to brain changes. So for example, enlarged perivascular spaces, microbleeds, small infarcts, and brain atrophy. Next slide. So using data from the COVID questionnaire and from the seroprevalence study, we're going to select cases and controls and then look at markers of cerebral small vessel disease, white matter integrity and myelin, brain functional networks, perfusion, brain structure with MRI, and then cognition with the CLSA neuropsychological test battery. Next slide. There'll be 328 cases with COVID-19, 328 controls, and each participant will have a brain scan, scanned twice 2 years apart, and the subset will have a third scan, which at 1 year in between the baseline and the 2 year. Next slide. So this shows the study design. So we are in the middle of scanning now. We've scanned about 280 of the 656 participants, so we hope to finish by April. Next slide. These are the sites that are involved. So they include participants in the CLSA study in Calgary, as well as these other 7 sites. Next slide. And we are collecting these data. In addition to the MRI and the CLSA cognitive test battery, we're also collecting questionnaires for anxiety and depression, because psychiatric symptoms have turned out to be quite common after COVID. Next slide. And this is the neuropsychological test battery. It consists of the Ray Auditory Verbal Learning, which is a verbal memory test, the mental alteration test, which tests executive function, animal fluency, which tests language and executive function, and questions on subjective cognitive decline. Next slide. And the neuroimaging protocol, will include T1 weighted, T2 weighted, FLAIR, SWI. Some sites will do arterial spin labeling, some will do myelin water imaging. And with these data, we can look at vascular lesions as shown in the bottom right, according to standards for reporting vascular changes on neuroimaging. Next slide. The data from this study we'll use to test our pre-specified hypotheses about vascular lesions in the brain in cases and controls. But then after that, the data will be made available to the scientific community according to CLSA data access policies and procedures. The website is shown here. So I hope many of you will access these data. Next slide. So in conclusion, the effects of SARS-CoV-2 on the brain are still being defined. We know in the hospitalized patients, they often have delirium, brain infarcts, microbleeds, white matter changes, atrophy. Particular parts of the brain connected to the olfactory system have been detected in the UK Biobank study. We know there is an increased risk for long-term neurological and psychiatric conditions, so we need to understand their basis. The CLSA COVID-19 study will be a source for neuroimaging, cognitive, and neuropsychiatric data on persons with and without history of COVID-19. Thank you.
Eric, thanks so much. So much to unpack there. One question that comes from Adam regarding the neurodegeneration of gray matter in the olfactory regions. Is there a likely return, or is it irreversible damage that you're seeing?
Oh, it's just an interesting question. They just have 2 time points, right? Like they've reported, and one of the advantages they have with the UK Biobank is their MRI sub-study is nested in a study where they had a previous baseline MRI before COVID-19 existed. So we know there was more, and changes in the regions occur with aging as well. So we know there was separation between the groups. And actually the degree of separation I did mention was greater in older people than younger people. You would need a 3rd time point. That's an interesting question. Like maybe they converge again. Maybe there can be some regeneration. We don't know. The MRI scans were done like months after infection, typically. You know, they weren't done at the time of infection. They were done after recovery. So I suspect the loss of gray matter is probably permanent, but I hope they're going to, and I don't know for sure, but I hope they will do at least one more follow-up MRI to see if this accelerated atrophy continues, ceases, or maybe if there's some regeneration. Thanks.
A question from Sarah, knowing that COVID-19 causes the long-term diseases in some cases, is there anything that you can do specifically, that we can do specifically to prevent these potential issues? You know, the increase in mood disorders, Sarah uses as an example.
No, that's a great question. And we're still like grappling with this new condition. And I don't know if the work that Catherine or Chris is doing is related to this. I'll be very curious to hear if they have an opinion on this. But I don't know. I think there is some increased risk according to the severity of COVID-19 infection, to my understanding. So like the critically ill patients tend to have more problems. So one thing we can do, obviously, is spread the word that vaccination is an effective strategy. It does not necessarily prevent all cases and transmission, but there's very robust evidence that vaccinated persons have milder cases of COVID-19. So I think that's probably the most important thing from a practical and preventive standpoint.
Some wise words to wrap your session, Eric. Thank you so much.
Thank you.
We'll double back with you as part of the panel at the end, but I'm going to pass it off to Preetma for our next speaker.
Excellent. Thank you, Jordan. So our next up is Dr. Catherine Lebel. Dr. Lebel's research uses structural and functional magnetic resonance imaging, MRI, to investigate brain structure and function in children. Broadly, her work is focused on understanding brain behavior, relationships in both typical and atypical brain development. Specifically, Dr. Lebel's work focuses on relationships between brain maturation and factors including prenatal environment, cognitive abilities, behavior, and mental health in typically developing children and children with developmental disorders. Welcome, Catherine. I'll pass over the stage to you.
Thanks very much. Just wait for my slides and then we can get going. Perfect. So Eric talked a lot about sort of direct effects of COVID on the brain. I'm going to talk about indirect effects of COVID on the brain, and specifically indirect via the stress that's induced by the pandemic, and then how that affects the infant, well, actually the fetal brain, and then ultimately the infant brain. So next slide, please. So prenatal distress is really common in non-COVID times, with estimates of about 20% of pregnant individuals experiencing depression and or anxiety. So for example, this plot shows the prevalence of people with clinically elevated symptoms of anxiety or depression in pre-pandemic meta-analyses. So about 20% is the estimate for anxiety and about 10% is the estimate for depression. And then we have the general US population, not necessarily pregnant, at slightly lower rates there. So this prenatal distress is concerning, obviously, for the pregnant individual. It's also concerning for their children because prenatal distress is associated with an increased risk of behavioral problems and later mental health problems in those children. So next slide, please. So I'm going to talk about the brain. And so just a very quick overview of a couple of the measures I'm going to talk about in the brain. So we can use diffusion imaging to look at structural connectivity in the brain, or the brain's wiring, the white matter. Diffusion imaging measures water movement within the brain. And because white matter is highly ordered, it creates ordered diffusion, which we can use to map out the brain's white matter connections. If you just click, we'll see. Next slide. There we go. These are an example of some of the connections we can map out within the brain, and we can query the properties of those connections. We can also use resting state functional imaging to get at brain function. And so here at the brain signal over time, and you can look at the time course of different brain regions and then correlate those regions. And when those regions are closely related to each other in terms of their time course, we would call those areas functionally connected. So these are the two primary ways that we in this work, and the two primary things I'm going to talk about today. Measure brain. So next slide, please. Next slide. I don't know if other people are having a delay, but I'm having a delay in seeing my slides. Can you go back one, please? There we go. Okay, and just click one more time back so we can see the start of that slide. There we go. Okay, so, oh, there we go. So depression and anxiety, as I said, affect about 20% of pregnant individuals, and they're associated with behavior changes in the kids. They're also associated with brain alterations. So this is a study we did several years ago. It was pre-pandemic, where we looked at depression symptoms in moms during pregnancy, and then we looked at brain structure in the kids. And we found that second trimester depression symptoms in moms were related to cortical thickness in the kids in an area of the right frontal lobe. Okay, that's the first scatter plot and little brain inset there, where higher depression symptoms in moms were associated with thinner cortex in their kids. This is sort of preschool age kids between 2 to 6 years old. We also looked at the white matter that connects to that cortical region and found that that white matter, properties of that white matter were also related to the depression symptoms. So higher depression symptoms in pregnancy were related to mean diffusivity or water movement in that underlying white matter. So click once here. There we go. And the interesting thing about this is that both of these properties, the cortical thickness and the mean diffusivity, decrease with age as a normal part of brain development. So what we're seeing when we see thinner cortex and when we see lower mean diffusivity is somewhat of a more mature pattern of brain development in these kids. So it's almost like these depression symptoms prenatally are causing these kids' brains to be a little bit more developed, like prematurely developed in these areas. And what's interesting about these areas is that these are areas that are related to later behavior and mental health in these kids. So we're seeing sort of a potential link here between the prenatal distress and the child behavior. Next slide, please. We see the same thing with functional connectivity. Here we looked at anxiety, prenatal anxiety, and we saw altered brain functional connectivity in the kids. This time actually reduced or weaker brain functional connectivity was associated with higher anxiety symptoms in the mums. And one thing that's important to note both in this anxiety study and also in our depression work is that most of these mums didn't have clinical levels of symptoms. Okay, these were women experiencing very typical ranges of depression or anxiety in pregnancy, not levels that would meet the clinical threshold for diagnosis. And yet we still see these associations between the level of symptoms and their children's brains. Next slide, please. So we see these brain alterations, and I've suggested that they might be linked to behavior, and we actually do see that. So here we looked specifically at a connection between the amygdala and the prefrontal area in the brain, which is a connection we know is related to emotion and behavior. And we saw that it was a mediator of the association between depression symptoms in mums and externalizing behavior in the kids. So things like aggression and hyperactivity. So this is really telling us that these brain alterations are a potential mechanism via which the prenatal distress can impact child behavior. Next slide, please. So we're thinking about all of this. We've been doing this research in the lab for a number of years, and then the COVID pandemic hit, okay? And obviously, we all know it has massively disrupted people's lives, but this has been especially true for pregnant individuals. They experienced a lot of alterations to their healthcare, so canceled appointments, giving birth without a partner there, unsure what the birth would actually look like. They're mostly women, of course, so they've had a disproportionate effect on their work. And also people who are pregnant are likely to have other young children at home. And we all know there were big disruptions to schooling and childcare during the pandemic. So really stress upon stress upon stress. And added to this, people have had reduced social support and reduced interactions with their typical support systems, with other new parents, those kinds of things. So it just becomes even more difficult sometimes to deal with that stress. So all of this has led to a very much elevated stress for pregnant individuals. And then we know, if you just click there, we know from all of this previous work that this is likely to have a lasting impact on the kids. So with this in mind, we launched the Pregnancy During the Pandemic study. And if you can just go to the next slide, please. This is a Canada-wide study where we recruited over 11,000 pregnant participants from across the country, starting in early April 2020, so right at the beginning of the pandemic shutdowns. And we recruited people over the course of about a year. People could participate in English or in French. We had representation from each province and territory, and this shows the demographics of our sample, which are somewhat comparable to the Canadian average, although people were more likely to have a higher education, had a slightly higher income, and were more likely to be married than the general population of pregnant individuals. Next slide. So the study structure involved an initial enrollment survey, which asked about depression symptoms and anxiety symptoms in pregnant individuals. It asked about life changes, particularly related to COVID, so changes in work or childcare or finances. It asked about some protective factors, things like social support, physical activity, and really all sorts of questions to really get a picture of what people were experiencing at the time. And then participants were invited to complete follow-up surveys monthly or bi-monthly through pregnancy. when they had their baby, they completed a short survey of birth outcomes. So, gestational age at birth, birth weight, those types of things. And then now our participants are completing postpartum surveys. So we have 3, 6, and 12-month surveys ongoing, and we're about to launch our 24-month survey. These postpartum surveys collect information on maternal health, as well as child health and child milestones. And click, please. We are also collecting brain imaging data at 3 months and at 12 months in Calgary here. And so that's what I'm going to mostly focus on, although I'm going to give you some results of the whole cohort first. So next slide, please. So I mentioned before, we're used to seeing levels of about 20% anxiety, 10% depression pre-pandemic and during pregnancy. The rates we see in our pregnancy during the pandemic cohort are about 2 to 3 times that. So almost half of our participants had clinical levels of anxiety during pregnancy, and about 1/3 of the sample had clinical levels of depression. So really massively elevated stress. We expected it to be elevated. We didn't expect it to be quite this elevated. Next slide, please. We also asked people about fear of COVID specifically. Okay, so obviously there's no validated scale for this, but we came up with a few questions to get at this. So how much do you think your life is in danger because of COVID? How much do you think your unborn baby's life is in danger? And how much are you worried that exposure to COVID will harm your baby? So people were moderately concerned about their own lives and their baby's lives, but a bit more concerned that COVID might harm their baby. And this was really before we had a lot of information about how COVID actually affects the fetus during development. Next slide, please. So some of the reasons for this stress were prenatal care changes. So we looked specifically at this, and prenatal appointment cancellations, changes to the birth support person. Often that was not being able to have someone at the birth who the mom wanted at the birth. And changes to one's childcare plan for the birth were all associated with higher risk of depression symptoms in the moms. Next slide, please. Now, on the positive side, we also found some factors that were sort of protective of distress. So women who reported higher social support, and this was generally social support provided by their partner, reported lower scores of prenatal maternal distress. So this is great, right? It's something that is easy to do. It's something that other people can help provide. And it's very encouraging that social support helps protect against those symptoms of anxiety and depression for these women. Next slide, please. So these are the sort of general birth outcomes in our cohort, which are pretty typical of Canadian births. Just click again and we'll get some other stats on NICU admission, preterm birth, and then of course a number of people experienced miscarriages or neonatal death. Next slide, or next click, please. Yeah, so a couple of these were associated with fear of COVID. So people who reported higher fear of COVID on those 3 questions I showed, their babies were more likely to be born a little bit earlier and have slightly lower birth weight. And then click again, please. And a number of these outcomes were also associated with cancellations to or disruptions to prenatal care. So the gestational age at birth, the mode of delivery, whether it was vaginal or cesarean, and then also NICU admission were all associated with those disruptions to prenatal care. Next slide, please. Now, this is the part that I'm really interested in, the brain imaging part. So we have data from 75 infants that underwent neuroimaging at 3 months here in Calgary. And we collected diffusion imaging to measure white matter connectivity, and we collected resting state functional imaging to measure functional connectivity in these infants. Click, please. And we looked at associations between prenatal distress as a combination of the depression and anxiety symptoms, and then metrics of structural and functional connectivity. And I think there's one more click here, yeah. And we controlled for a lot of the covariates that we know are associated with various parts of this, including maternal education, postpartum distress in moms, household income, infant age, and infant sex. Next slide, please. And what we saw were associations between that prenatal distress measure and structural brain connectivity. So here I'm showing on the left is the uncinate fasciculus, and on the right is another amygdala for prefrontal tract. And both of these had relationships between that prenatal distress and these children's brain connectivity. So on the left, we see higher maternal distress associated with higher fractional anisotropy in these infants. And then in the right plot, we see lower mean diffusivity associated with higher prenatal distress. And so these are very similar relationships to what we'd observed in our cohorts pre-pandemic, both in terms of the direction of associations. We're seeing sort of a premature pattern of brain development here. And in terms of the area where we find the differences. These are connections that we know are involved in later mental health, emotion regulation, behavior in the children. Next slide, please. Interestingly, some of the metrics we looked at were moderated by infant sleep. So here we looked at brain efficiency in a limbic frontal network here, and we see that higher prenatal depression scores were associated with reduced brain efficiency in infants, but primarily in infants who had less overall sleep. In infants who got lots of sleep, this association wasn't there. So really interesting in terms of potential protective factors, right, on the infant sleep side postpartum. Next slide, please. We also looked at brain function, and here we also saw associations where higher maternal distress was associated with weaker brain functional connectivity. So again, this is similar to the findings we had seen pre-pandemic, but this time we saw that it was moderated by social support. So women who reported high social support, their distress was not related to their infant brain's functional connectivity. But women who reported low social support did have this association between higher distress and weaker functional connectivity. So again, really interesting sort of here in terms of potential protective factors, and quite promising, I think, for potential interventions. Social support is an easy intervention. You facilitate safe places for people to meet and connect, and things are available like couples therapy to improve partner relationships. So really encouraging, I think, that social support not only supports the pregnant individual, but potentially has the potential to support that infant brain development as well. Next slide, please. And as I've been sort of harping on throughout, these brain areas are quite relevant because we know they're related to later behavior and mental health. So I think what we're seeing are potentially the early beginnings of something that might predispose some of these children to later behavior and mental health challenges. But of course, these kids are developing, so there's a lot of time to support them and help them as they grow. Next slide, please. So in summary, the COVID-19 pandemic has led to very high stress in pregnant individuals, and this stress is associated with both brain structure and brain function in their infants. Association is moderated by social support and by infant sleep, so both of those might be potentially protective. Next slide. And so I think what's really critical here is that it's really important to support perinatal mental health. And that goes not just for the pandemic, but after as well. We need to better understand their trajectories and how they're going to grow and behavioral changes in them. And I think there's a need to continue to study potential risk and protective factors to better understand where these associations might be the most prominent. Last slide, please. And so finally, I want to thank particularly the co-leads of this pregnancy during the pandemic study, Leanne Tomfor-Madsen and Jerry Giesbrecht. I want to thank all of the fantastic people who've worked on this study, the coordinators, the trainees, all of our volunteers. I want to thank our funding sources, and I want to thank you all for your attention today.
Thank you so much, Catherine, for your talk. That your work is absolutely fascinating. I myself had my baby boy just before lockdown in January. So I was one of the lucky ones, but I can certainly empathize with your work. And it feels like the theme of today has been, nurturing our bodies with micronutrients, with sleep, with rest, with connection, is really important across the lifespan. So really meaningful work. We have a few questions here for you. Svenja is asking, how difficult or challenging is it to do the imaging on infants, and how successful are you in gathering this valuable data?
So scanning is always challenging. It's especially challenging in little kids. The infants are actually easier than the toddlers and preschoolers, which we've been doing for a number of years. So the infants, scanning infants, we get them to feed, and then we wrap them up, swaddle them tight, bundle them in a bundle bag, and hope they fall asleep, and then hope they stay asleep. And when they're asleep, they tend not to move very much, which is good. So we can usually get about half an hour to 40 minutes of imaging time in there. They're not all successful. We're probably getting decent images on about 2/3 of the kids that we bring in. And then as they get closer to a year, it gets a lot more challenging to get them to fall asleep. They're not as willing to fall asleep wherever they are.
Interesting. And do you have any words of wisdom to those in the audience who have friends and family who are those who are pregnant that are experiencing concerns about the COVID-19 vaccination? Is there any correlation or concern with regards to that?
So we've asked our women if they're getting vaccinated. Most of them are, of course. The data really strongly supports vaccination, even in pregnancy or while breastfeeding. There's really no reason to think that that's a risk of any kind. And in fact, it's likely highly protective, because we now know that getting COVID while pregnant is a risk to preterm birth and potential other complications. So we haven't looked at that with our work specifically, but the evidence is very strongly supportive of that.
And did getting the vaccine in that population, did it provoke any further anxiety for folks in the study?
So that is a good question. We didn't ask that specifically. Keep in mind, we launched our study right away. So the vaccine wasn't available for most of our participants while they were pregnant.
Right.
I think pregnant women were one of the higher risk categories, but I think we were still talking about March 2021, kind of when most of them would have been eligible for vaccines. And we recruited from April 2020 to April 2021. So most of them would have been pregnant before the vaccine was available.
I see. Okay. Any final remarks? Anything that you'd like to add before we move on to the next presenter?
No, I'm happy to carry on. Thanks for the opportunity to speak today.
Thank you so much, Catherine. I'll pass it over to Jordan.
Thanks, Preema. Thanks, Catherine. Our final presenter of the day, two presenters. I would like to introduce both Dr. Christopher Power and Andrej Roczkowsky. Christopher Power is a neurologist and a professor at the University of Alberta and holds the Henry M. Tupen Chair in Neurocognitive Disorders. He received a Bachelor of Science from the University of Toronto and an MD from the University of Ottawa, with subsequent training in internal medicine and neurology at McMaster University and the University of Western Ontario, respectively, as well as postdoctoral fellowships at Johns Hopkins University and the National Institutes of Health in Neurovirology. Dr. Power is a physician scientist who studies the mechanisms of neuroinflammatory diseases and their treatments. In addition to directing his translational laboratory, the Brain Power Lab, he is also an attending physician in the University of Alberta HIV, MS, and Neuro-COVID clinics. He is an author on over 200 peer-reviewed publications, 20 book chapters, and he holds multiple grants from CIHR and MS Society of Canada. Andrej Roczkowsky is a first-year Doctor of Medicine student at the University of Alberta. He obtained his Master of Science in Pharmacology at the University of Alberta, studying ischemic heart disease. His research interests include heart disease, multiple sclerosis, and the effects of COVID-19 on the brain. Gentlemen, thank you for joining us.
Thanks, Jordan. If I could just have the first slide. Great, thanks. So before I begin, I just want to make a couple of disclosures on behalf of Andrej and I. We're funded by the CHR and also the MS Society, and also the Weston Institute. And the other thing I'd like to acknowledge is that I'm speaking from the traditional territories of the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, and Ojibwe Saulteaux nations, and for also known as Treaty 6. Next slide. Thanks. So I'm going to give a brief overview of perspective of viruses and brain diseases. I hope this won't overlap too much with Eric's talk. Then we'll talk a bit about potential disease mechanisms. Andrej will address that specifically, and also and at the end we'll discuss future studies of COVID-19 in the brain. Next slide. So I think we all know that we're in an era of emerging infections, and that this is going to continue to be an issue for all of us going forward. There are multiple factors that contribute to emerging infections like COVID-19. Susceptible populations certainly that's changing as people age. We also have altered interactions between humans and other species. Climate change is unquestionably a contributor, and then of course individual infectious agents are evolving and changing. There's always an element of chance as well in this, but certainly as illustrated down in the left-hand corner of this slide, there are multiple infectious agents that start out in animals and then switch to humans, and they evolve during that time. Medical practices have also influenced emerging infections. And finally, perhaps one of the biggest factors that contribute to emerging infections is our ability to travel quickly to right across the globe. And when you think of back 100 years ago, it would take months or 6 months to go from Europe to North America and across North America. Now we can do it in merely hours. So the potential for transmitting infections is immense. So we have to keep all of these variables in consideration when looking at emerging infections. Next slide, please. So this is a slide that I update, and you can see I've been updating it for a long time, since HIV emerged in 1981, but you can see over time that many infections have come to recognition, and most recently SARS-CoV-2. But we will see more. This isn't actually a complete list. There have been others that have emerged during this time, but I think it's very telling that we are facing very different times from what our parents and grandparents faced, and that we are going to have to face new infections ongoingly, and we'll have to deal with them on a very specific basis, depending on the infectious agent. Next slide. So we've known about coronaviruses for a long time. Eric gave a very nice overview of SARS-CoV-2, which is the infectious agent responsible for COVID-19. But of course, we know there was SARS-CoV-1 back in the early 2000s. But even before that, we knew about other coronaviruses. In fact, coronavirus OC43 is known to infect the brain in humans and has been associated by with MS by Canadian investigators at the University of Montreal. There's an animal model of MS that depends on a coronavirus. And of course, more recently, viruses like MERS in camels in the Middle East have been associated with respiratory disease, but in a few cases also neurologic diseases. And so there's a tremendous capacity for coronaviruses to cause neurologic disease and also to jump species as well. Next slide. So the infectious agent that we're obviously most concerned about is SARS-CoV-2 today. And its origins remain murky. We know that it's certainly viruses very much like it are present in bats, but it's not clear whether there was another species it went into before going into humans. What is very clear, I think, is it's not a escaped agent from the labs in Wuhan. I think the epidemiologic studies from people like Eddie Holmes and other groups suggest that this is actually from the wet markets in Wuhan and perhaps other markets as well. And that's how it got into humans. Eric's done a really good job on discussing its receptor, but it uses a very distinct receptor to get into cells, and of course produces tremendous respiratory disease, but of course also neurologic disease. Next slide. So right from the outset, we knew there were neurologic illnesses associated with COVID-19. The sense of loss of smell and altered taste, so-called anosmia or dysgeusia, but confusion, stroke, seizures, and also the peripheral nervous system is affected as well. And then there are other syndromes that tend to be more associated with sort of post-viral illnesses, something called acute disseminated encephalomyelitis, so-called limbic encephalopathy, and indeed we've seen these syndromes. These are not uncommon. We see them on the wards. In the sort of peak last fall, when I was on call, we saw patients that had very atypical neurologic syndromes that are almost certainly associated with COVID-19 infection, which looked like they might be so-called autoimmune disorders. And so this is a very real issue, and we don't fully understand the mechanisms yet. Next slide. The other thing that was alluded to is after the acute episode is the so-called long haul syndrome. And this has garnered a lot of attention because it affects maybe as many as 25% of people. It resembles in some ways something called chronic fatigue syndrome or myalgic encephalomyelitis. There are many shared aspects or features of that. And and of course that is problematic because we don't really understand what causes chronic fatigue syndrome, but phenomenologically, it looks very similar to long haul syndrome. Next slide. It's quite a constellation of symptoms and signs that you see in the long COVID or long hauler. It's also, that's the technical name is post-acute sequelae of COVID-19. But there are a variety of features, all attributable to neurologic dysfunction, cognitive disorders, mood disorders. Autonomic dysfunction is so difficulty with blood pressure, rapid heart rate. Pain syndromes are hugely common. I see many patients in our post-COVID clinic that have chronic neuropathies and myalgias or muscle pains that seem persistent, and also nasty headaches as well. And of course, there's exercise intolerance. Next slide. This is a slide, the top three panels show that there's a high risk of psychiatric disorders after COVID, anxiety, mood disorders. And then below is data from one of our neurology residents who's got a prospective project looking at the risk of different neurologic syndromes in patients who've had COVID or have not had COVID, so they have controls. And it looks like the risk of dementia as well as seizures is higher amongst those patients who've had COVID. This is actually data from Alberta, about a cohort of about 200,000 patients. So, and there is certainly an increased risk of these neurologic disorders, which recapitulates to some extent what Eric said earlier. Next slide. What are the underpinnings of these neurologic disorders remain uncertain. The question of virus in the brain, Andrej will deal with that directly. Systemic inflammation is a big issue. Autoimmune disorders, unquestionably, it's a factor. Just inflammation in the brain, we'll show you some data. And then damage to blood vessels, Eric's dealt with very well, and certainly that's a major issue as well. So next slide. So I think at this point, I'll turn it over to Andrej, and he can describe some of the experiments that he's been doing in our group. So Andrej, over to you.
Thank you, Dr. Power. So we studied the mechanisms of COVID-19 induced brain dysfunction and asked two research questions. First, is SARS-CoV-2 present in the brains of people with COVID-19, as well as in our animal model of COVID-19? And second, is inflammation present in the brains of people who have COVID-19, as well as in our animal model? And on the next slide, we received brain tissue from 10 individuals who passed away from COVID-19 and focused on 3 brain regions. The first was the olfactory bulb, which is critical for smelling. We also looked at the cerebrum, which is responsible for higher order thinking and information processing, as well as the brainstem, which controls the basic subconscious parts of life, such as breathing. And we looked at the levels of SARS-CoV-2 RNA present in these 3 brain regions, as shown on the graph on the right. On the very far left of the graph is a positive control, which is lung tissue from a COVID positive patient. And we only detected small amounts of SARS-CoV-2 RNA in only a subset of patients in these 3 brain regions. So the majority of patients had no detectable virus present in their brains. On the next slide, we then wanted to confirm the presence of SARS-CoV-2 in one of the patients who had detectable virus in their brains. And we did this by a technique called immunofluorescence microscopy. We detected 2 different SARS-CoV-2 proteins, NSP3, which is in yellow, and spike, which is in green, which were present in the brain of a COVID positive patient. And importantly, we did not detect either of these 2 proteins in the brain of a COVID negative patient. And in red on the left is GFAP, which stains astrocytes. And astrocytes are a type of brain cell that support neuron function. And we saw quite a bit of COVID protein colocalized with the GFAP signal, suggesting that these proteins are present inside of astrocytes. Next slide, please. And inflammation has been reported in the brains of patients with COVID-19. So this figure here shows localized inflammation in brain tissue from a COVID-19 positive patient. On panel C is CD68 staining, which stains brain macrophages and microglia, which are immune cells. And so this figure shows localized inflammatory lesions present in the brain of a COVID-19 patient. And next slide, please. We then looked at the expression of inflammatory genes in all 10 of our COVID patients. And we saw an increase in the expression of inflammatory genes in all 3 brain regions of the patients who passed away from COVID-19 compared to non-COVID-19 patients, particularly in a inflammatory signaling molecule known as interleukin-8. And we also saw an increase in the expression of genes involved in cell death compared to non-COVID patients. Next slide, please. We then wanted to confirm our findings in a hamster model of COVID-19. So we infected hamsters intranasally with SARS-CoV-2 and collected their brains at day 4, day 7, and day 14. We then looked at viral load in the olfactory bulb and the cerebrum. And in the olfactory bulb, we were able to detect virus at day 4 and day 7, but the virus was cleared by day 14. And we did not detect any appreciable levels of virus in the cerebrum. So on the next slide, we then looked at the expression of inflammatory genes in the olfactory bulb of the hamsters that were infected with COVID. And we detected a significant increase in inflammatory genes at days 4 and a little bit at day 7 as well, but not at day 14. And again, we also detected an increase in genes involved in cell death compared to uninfected hamsters. Next slide, please. Then looking in the cerebrum, we did not see any inflammatory response at the earlier time points of day 4 and 7. But at day 14, we saw a significant increase in the expression of inflammatory genes, as well as genes involved in cell death. And so it looks like in the cerebrum of infected hamsters, there's a delayed inflammatory response. And next slide, please. So to conclude, SARS-CoV-2 is present in the brains at very low levels, but there is abundant inflammation in the brain during COVID-19. And for our future directions, we want to determine the clinical risk factors associated with developing COVID-19 induced neurological disorders, as well as investigate novel therapies to prevent COVID-19 associated inflammation and cell death in the brain. And next slide, please. So I'll end with a quote from Dr. Fauci. In the fight against this pandemic, we must lead with science, and a key piece of our ongoing work is communicating consistently. So on the next slide, I'd like to thank everybody that contributed to this research. If you could click again to get the names on the slide, please. I'd like to thank Dr. Langdon and Dr. Joseph from the University of Calgary, as well as everybody from the University of Alberta, Dr. Doss, Dr. Hobman, Dr. Lamonta, Dr. Evans, Dr. Lamb, and Dr. Backel, as well as everybody from the Power Lab, Brittany, William, Aaron, Candice, Jason, as well as Natasha. And thank you to Campus Alberta Neuroscience for inviting us to speak. Thank you.
Thanks, Andrej and Chris, very much. You know, I think where I want to go right away, no one has listed a question in our chat, but Andrej, I think it's a great quote that you share, and it talks a lot about the communication of science, and something that the broader science community has seen. It is something we're up against coming through COVID. It's something, from my background as a meteorologist, our community of climatologists and meteorologists have struggled for years with climate changes. How are we not communicating effectively? So how does that communication piece fit into your team and colleagues, as well as it goes to really complicated science, but making it accessible to folks who are asking questions and trying to learn and get information that isn't fed through the media, but communicated effectively through academia?
That's quite a question, Jordan.
Sorry.
Maybe I'll defer to my junior colleague. Maybe you can answer that.
I mean, to narrow it, do you have plans in place, or are you engaging certain teams at the U of A to help in the communication piece?
Yeah, we operate through, we have an advancement office, so we communicate our results through our advancement office, and they, I think getting the message clear and straightforward is really imperative. Dr. Fauci's been, I think he's been really outstanding in making sure that people really understand that it's, you know, it's critical. Vaccination is critical. This is not a conspiracy. That and I think so. Got to be sure that you keep your facts and your values separate here when communicating your data, and I think that's a good way forward. But yeah, we verify, or before we send stuff out, we're careful to run it through our communications office and advancement office in the faculty. I guess I hope that answers your question.
Yeah, I mean, absolutely. You know, I'm coming at it from the background of being both a meteorologist, a climatologist, and a communications expert. So that's very near and dear to me. And it's almost been watching scientific colleagues come on board to the struggle we've had for 20 plus years in climate change to go, oh, now here's another group of scientists in medicine that are seeing that struggle and trying to communicate something effectively. It's very important. But you're right, universities across the board have great teams to communicate. So it's nice to hear you stand up for that at the U of A as well. I won't beleaguer that point. Lori asks, could you clarify if the patients included in the study were fully vaccinated or not?
Yeah.
Yeah.
Do you want to answer that question, Andre, or do you want?
So I believe quite a few of the patients, their samples were collected prior to the vaccine being available. So I don't believe the majority of our patients were vaccinated, but I think Dr. Power can clarify if it's actually all of the patients that haven't received the vaccine.
No, Andre's right. You're exactly right. But I will comment, though, in our long COVID clinic, we are definitely seeing more patients who were not vaccinated. The vaccination seems to have a beneficial effect in that respect. So, which is reassuring.
For sure. We'll do one more question, then we'll open it up to the panel. We'll invite our other speakers back. Anna asks, what are the risks to human brains to reopening Alberta's post-secondary institutions from lockdown when other countries are going back into lockdown from COVID-19?
Right. Yeah, that's a really good question. Maybe I should leave it to Catherine since she's more familiar with developing brains. But I will say that I'll volunteer this, that I think we should be very cautious going forward, and that we should be driven by the science and not the ideology. I'll leave it at that.
Fantastic.
Maybe wants to weigh in on that as well.
Let's invite Catherine and Eric back, and maybe we can leave the floor open to start if Catherine or Eric does want to weigh in on that conversation first, or we can move on and direct a new question. Anything to say on that? Just sort of the pace of bringing large organizations back together?
It's hard to say. I think in some ways the conversation, which one side says follow the science, the other side says do your own research. In other words, it's fake science, do what feels right kind of thing. I think they're both sidestepping the question, which is a question of values, actually. I mean, we should make science-based decisions, understanding the kind of the benefits and the risks of different choices. But in some people's value system, it would be an acceptable choice not to be vaccinated, right? Even though I know that's not maximizing the chance of having a good brain health outcome. I mean, that is an objective fact. But if the value of having the choice of the vaccination exceeds that medical risk, that's a question beyond science that gets at like kind of values. And I'm not a sociologist or philosopher, so I guess I'm going to plead, or a politician. I guess I'm going to plead a bit of ignorance here, but I would like us to see, rather than implying that the science tells us what our values should be, which I think is incorrect, or just ignoring the science altogether, which unfortunately too many people are doing. I would like to see us incorporate the science into our values-based decision-making.
Catherine, anything to add to that?
No.
That's fair.
Yeah.
Very
That's
fair.
what I should have said.
I'll open up sort of a first question then to everyone, and maybe Catherine, you can take this one first. Are there healthy brain habits that can be used to mitigate the risk of COVID-19 to the brain, but maybe even more broadly, other viruses that we know about? Just healthy brain habits in general that people can adopt.
Sure. So can you hear me? I'm cutting in and out a little bit. So
Yeah, we can hear you good.
I can.
are you able to hear me?
Yep.
Okay. So maybe I'll take that from my sort of indirect brain effects from our study, but brains are always malleable, especially pediatric brains, but other brains too. And I think there's lots of stuff we know that's good for brain development, and that stuff remains good for brain development even during COVID. So positive interactions with caregivers, good learning environments, supportive, stable homes, those things are all are good always for kid development. So I wouldn't say necessarily from my perspective with the indirect effects, there's anything particular we should be doing that's different, but just that we should be making sure we still support safe, supportive, positive environments for kids throughout.
I think it's an interesting point, and maybe it falls back to, Eric, what were you were saying about striking a balance too, and how we approach a return to life with COVID. So maybe Chris and Eric, you could weigh in on that thought process of, you know, healthy interactions as well. Catherine's talking about developing brains at a very young age, but it's that social interaction that is another piece that is healthy for our brains and may offset some things. Is there a comment to add to that?
Maybe I can comment on this a little bit first, maybe then you can go, Chris. That we see anecdotally, you know, we had a lot of comments from patients seen in our dementia clinic and their family members or care partners, you know, that the social isolation necessary, right, to reduce risk of transmission in the early waves, you know, had adverse effects on people's cognition and mental health. Often the cognition declined in the patient and the mental health declined in the care partner. We use this term called cognitive reserve in like aging and dementia research, which is the idea that brain healthful things can like make your brain more resilient to other challenges or pathologies. The kind of neurophysiological and basis of reserve are some of these concepts that Catherine referred to, you know, the brain is plastic, and by enhancing and creating robust networks and maintaining them, it may, you know, that may be the physiological basis of this resilience. And that has been challenging because of COVID, you know, with less opportunity for interactions, you know, between humans. So that's something that's partly been replaced by these Zoom calls and these webinars, and in some ways it's been enhanced a little bit, right? Like by not having to physically travel to interact with people, but you know, there's been a downside to it too. So I don't know what the right answer is. I don't have a grand solution for the right balance, you know, to kind of maximize the utilities here in terms of balancing social interaction with actual risk of getting your body infected and the primary secondary effects on the brain. But it has been a challenge, right? Like kind of dealing with some of these aspects of the social isolation.
Chris or Andrej?
Andrej, why don't you have a shot at it?
Sorry, could you repeat the question?
You know what? I want to form it from your perspective, Andrej, being in a class setup as well as future MDs. What does that look like, coming together, sort of studying all of this still, but wanting to also in person complete your education in a proper manner? Yeah, what can you see as sort of the best outcome of still nurturing that social aspect of your brain in protecting yourself in a healthy way?
Yeah, I think that's been a really difficult part of COVID, especially from a student's perspective, and everybody going to school. Being isolated has impacted our education to some extent. So I think it's really important to maintain social contact with our peers as much as we can through conferences and virtual meetings like this.
Do we have any hypothesis on how long into the future we'll be continuing to deal with the impacts of COVID-19 on our brain health, whether it be through, you know, with those who have experienced COVID-19 and struggled with the symptoms, or in women going through their next pregnancies, experiencing post-traumatic stress? How long into the future are these impacts going to last?
So I can take a shot at that, Pridmarthi. I think there are two sources of information. One is that we have known about these post-viral illnesses for a while. So people working in developing countries, particularly in the Caribbean Basin, South America, where they have a lot more experience with viruses like dengue, chikungunya, Japanese encephalitis virus. Those, you can get very ill, not necessarily you can get brain disease, but you can get very ill. And those people are often left with a post-viral illness that looks very much like long COVID for months. But the good news is people tend to recover from them. And actually, that's what I see in the post-COVID clinic, the long haul clinic. People are for the most part, particularly the symptoms like fatigue, palpitations, shortness of breath, and anxiety, that diminishes with time, which is good news, actually. I was concerned, Eric showed some data, and we have some data, that the incidence of dementia and seizures goes up after COVID. I have a sense that COVID uncovers some pre-existing problems, and maybe Eric, you can speak to that a little bit. But so there are some disorders like dementia, which I think are accentuated or brought to the forefront as a result of COVID.
Yeah, I'd agree with you fully about the comment about dementia, Chris. We see this unmasking occur in the context of major surgeries like coronary artery bypass grafting,
Yeah.
or a hip fracture, right? And delirium around the time of an illness or hospitalization, it just in general, like, is known to be a fairly strong predictor of increased risk of future dementia. We're going to need more data to look to see whether this risk is just around the time of the illness or whether it persists. There's still ongoing debate about whether there are biological underpinnings like interactions of anesthesia with the brain in the context of surgery, for example. But some of it may just be in a brain with less reserve, undergoing just a general strain, that it kind of unmasks the fact that things were just barely compensated before. And now people recognize that, in fact, there has been a cognitive disorder for a while. It's just the stress of an acute illness has made it impossible to ignore, that fact. Yeah.
I want to double check in our raised hand section. Anna Wilson, you've raised your hand, but I noticed you also had something in the chat. So I don't know if we would be doubling back on a question. Anna? Okay, we'll move into Blaine's question. Blaine says, I'm speculating viruses like COVID will continue to be on the rise into the future. Is there a discussion in each of your fields about how to better deal with the next pandemic?
Yeah, I can speak to that a little bit. The federal government, so this has had sort of a long history of the federal government being interested in pandemic preparedness, dating back to the early 2000s after SARS-CoV-1. In fact, at that time, there was an initiative. I was involved with it because I was very involved with Canadian Institutes of Health Research and the Infection and Immunity Institute. That there was an interest that diminished for several years, and then in the last 3 or 4 years, there's been a heightened interest. And right now, the government is announcing a big pandemic preparedness across the country. The big players that I know of right now will be at McMaster and VITO. That's the Veterinary Infectious Disease Organization at University of Saskatchewan, but an effort to sort of lay the groundwork so that we are better prepared going forward. But I think we're seeing it in other spheres. The extraordinarily rapid delivery of vaccines. I mean, this was unbelievable that we could get a messenger RNA vaccine in months. It used to take a decade to get a good vaccine, and so we're going to see that. So will be better prepared for the next time round because there will be another similar event coming our way. So that's some practical. I don't know about brain health per se. I guess we better work on our cognitive reserves in the meantime.
Yeah, I think that's one way to prepare. Healthy diet, stay cognitively active. Be screened and treat vascular risk. And it's never too soon to start. Living a healthy lifestyle. You don't have to be as old as I do. You can be a graduate student and walk the walk as well as talk the talk. I found your presentation really interesting, Chris. I hadn't thought about it. I think there's just generally a tendency. I'm skeptical to say, oh, things are changed and they're worse now or better now than ever before, because there's a bit of a bias to thinking our own time is unique. But your slide showing all of these emerging infectious illnesses, and of course the penetration of humans into every habitable corner of the world and being in contact with different animals and the risk of zoonoses, you make a pretty compelling case that we should keep up our preparedness better than last time. Probably there's mostly graduate students here, so you may not have been around during during the SARS outbreak, which hit Canada hard in Toronto. And I know from medical colleagues there, it shut down Toronto's health system just in the same way that the current pandemic did to the Canadian and global health system. Like the hospitals it was very difficult to function. And SARS was scarier because the mortality was much higher. Thankfully, it was less transmissible, so it could be stamped out. And the preparedness in terms of these scientific techniques and the vaccinations was really exciting for me to see. But as a clinician and healthcare worker, it was, on the other hand, very scary and really we didn't have basics though. We didn't have gloves, right? Or like masks or anything at the beginning of COVID-19 pandemic. And I hope we can be prepared with some of these basics for the future. I'm sure that's going to be part of the response. The problem is in 10 or 15 years, when all the gloves are about to expire, when the plastic is snapping and breaking, are we going to have the discipline to maintain and spend the money to maintain the preparedness? I hope so.
I'm curious to know how the pandemic has positively impacted your guys' research agendas and removed barriers for you, and hopefully has removed barriers and expedited some of your work potentially for the years to come.
Well, it's mostly sucked, I guess I can say, to be honest, right? But there were some things that changed, right? Like we got over the barrier to using video conferencing software to interact with our research participants and to consent them remotely and to do cognitive assessment remotely. So in some ways it brought us in closer contact. We still need that human, physical contact too, but in some ways it brought us in closer contact suddenly and removed some of those barriers. So, but I'm curious what Chris and Catherine would respond as well.
For me, it launched our study, of course, which is it's a natural experiment, right? So, nobody would ever have wished for it, but it's a very unique opportunity to study some of the things that I had been studying before the pandemic, in terms of stressors and life changes. So it's, from a research perspective, it's been very fascinating to study something like this that is such a large stressor and such a global stressor for people.
So I guess from my perspective, it's a little different. It's a bit more granular. And Andrej, you can speak to this, but our team became very tight as a group. Right, Andrej? That really changed the dynamics within my research group, and it became kind of I think, correct me if I'm wrong, Andrej, but people wanted to come to work. We wanted to be engaged in this somehow and try and make a contribution, but we naturally pulled together. And I can remember at the beginning, March 13th, 2020, it was really scary, and people just responded, and it's actually put a very positive, there was lots of anxiety, of course, but at the same time, it really enhanced teamwork. I don't know, Andrej, do you want to comment? You were in the lab for a lot of this.
Yeah, I would agree. Having the exemption to keep doing research really felt like an opportunity, and what's the word? It was like almost like a pleasure to keep working, I guess. I can't think of the word right now, but because we had to stagger shifts too, it did really enhance our collaboration within the lab. We each worked on each other's projects a lot more and had to take over when somebody had to leave and somebody else had to fill in. So from that point of view, it was quite collaborative. At the same time, though, it was a little difficult to have to break up, all our experiments because we had to stagger shifts. But
yeah, I've heard overall, though, that the pandemic has helped reduce a lot of barriers in terms of funding and just some of the loopholes that have to be jumped through in all industries, including research and innovation, particularly getting innovations to market a lot quicker. which has been a real blessing, I think, that this time has brought upon us.
Can I say something? And this is political. It's been pretty rough at the universities in the last 2 years. I might as well say it because maybe everybody else on the call is too polite, but with major cuts to the universities, approaching 30% cuts to our budgets, I'd have to say it's been very, very challenging, and I'm very impressed by my colleagues that everybody's risen to the occasion. I think that needs to be said and said again because, yeah, I'll leave it at that.
Certainly a tremendous impact of the funding cutbacks, especially on such important work. Catherine, I'm interested just in a follow-up to your work there. Are there any studies, or is there a study going on with regards to just the development of the infant and toddler brain during these times, during post-pandemic times compared to pre-pandemic? Is there an impact there at all in the way that these children are being raised and how they're navigating through this mentally and emotionally?
Yeah, there are actually quite a few studies that were started before the pandemic, right, of brain development, and that are continuing to collect data during the pandemic. So you get this unique pre-post sort of sample with similar demographics from a similar area, those kinds of things. So I don't think there's a lot that's been published yet on that, but there are a lot of studies that have that kind of data, and that we'll be looking at it in the coming years. And I think it's really interesting. It's really important. It's going to be hard to tease apart what the contributing factors were. Was it stress related to the pandemic? Was it a lack of social interactions? Was it, lack of education opportunities when schools were closed? Like what is it impacting the kids? I think that'll be something that's very challenging to figure out. But I think we will start to know more in the coming months and years about just overall development and whether it looks different in kids. I mean, most kids are going to be totally fine and totally normal, just like they would have been before the pandemic. That's the reality of the pandemic, I think, is that the vast majority of people are going to come out of it just fine. But it's the people who aren't that I think we need to figure out how we can support and help. There are going to be people with long-term consequences of various kinds for various reasons. And that's where we need to put our effort. Most people will be fine, but it doesn't mean we should ignore the people that aren't.
Oh, Jordan, we can't hear you. Nope. Any final words, any final remarks that any of you would like to share that you didn't have an opportunity to do so?
Well, thanks for the opportunity to present our data. I think we're always happy to show our own data. That's nice. And for taking an interest in all our efforts. I think we've both Eric and Catherine, as well as Andrej and I have had to pivot in response to this, and it's wonderful to see such outstanding productivity and great data. So
thank you so much for everyone in sharing their time with us. Thank you for the day. We really appreciate your sharing of knowledge. Jordan, are you back on? No, we can't hear you. So I think we will take a pause now. We're just waiting for Dr. David Park to join us. I think he might be having some technical difficulties. So we are just waiting to get him connected. Just a sec. We can go to a bit of a 5-minute break and reconvene and for our closing remarks by Dr. David Park. Thank you, everyone.
Thank you.
Thanks.